The alternate pathway involves the spontaneous hydrolysis of C3 to form C3H2O, leading to the eventual deposit of C3 convertase C3bBb onto microbial surfaces, which leads to any of the three complement outcomes. The key molecules involved are factor B and D, properdin, and of course C3. This was the second complement pathway to be discovered. Because this pathway begins spontaneously, the immune system has developed measures to stop it from happening in the absence of a pathogen. If C3b, cleaved from C3, does not rapidly form a thiol ester bond with the pathogen, the complement cascade stops. Properdin accelerates the alternate path to increase the complement response, and factor B can also start the alternate pathway if needed. The alternative pathway is a key path in the complement system as it allows for an effective, localized, and strong complement responses in the presence of a pathogen. One of the outcomes of the complement pathway is the opsonization of the pathogen. This pathway occurs when the pathogen has C3b receptors which C3b can bind to. This makes it easier for other immune cells like macrophages to engulf the pathogen and destroy it. Macrophages have complement receptors like CR1 which can bind to C3b, which is bound to bacteria. This is how C3b helps direct macrophages and other immune cells to pathogens to destroy them. Another protein in this complement pathway, C4b, also plays a similar role in initiating opsonization. This outcome is very effective against extracellular pathogens as it helps target effector immune cells to the pathogen quickly to kill them and prevent further harm to the body. The C3 convertase step is the key step in all complement pathways and outcomes. The key portion of the C3 molecule is the thiol ester bond in the C3b component. This bond interacts with the pathogen surface, which tags it so that one of the three outcomes can occur to or around the pathogen. This step is essential because it bridges the activation of the complement system with the other effector outcomes. Within the three activation pathways and effector pathways, other molecules interact with C3b, such as C4b, C5, and C2. Overall, this step is extremely important in making the complement pathway an effective measure against pathogens in the body.
Fear not, brave soul, for your body does have another method available if the classical pathway of complement cascade activation has been compromised, and it's the lectin pathway. The lectin pathway takes these things known as MASPs, they're associated serine little pieces of protein, and basically instead of using the classic antigen-antibody complex to adhere to the invading bacterial cell, they use a bunch of proteins collectively known as collectins, and among these, the one that takes the top priority is the mannose-binding lectin protein. And once adherence is done, it goes through the same exact steps as the classical pathway. The C2 and C4 proteins are cleaved into C4b and C2b respectively, combining to form the C3 convertase. This convertase then allows for opsonization of the invading bacterial cell. And there you go, you won't take any L's with the lectin pathway.
The classical pathway is one of the three pathways of the complement system. It kills pathogens or anything that causes disease, such as bacteria. It aids inflammation, lets the immune system know what needs to be eliminated, and after everything, it removes the pathogens from the bloodstream. The major players of the classical pathway system are, of course, the microbes and the antibodies that attach to it. So how does it all work? Well, first off, the antibody attaches to the antigen. From there, it breaks off into multiple different proteins. It's a bit complicated, but the gist of it is, these protein fragments will attach to the cell membrane of the pathogen, essentially causing it to swell and burst, which is what kills the cell. Thank you for watching our pathways. We hope this was informative.